I know from Carl that the two of you go a long way back. And the title of his talk is inspired by Carl, Exploring the Microbial Dynamics Within. Larry.
Well, thanks very much. It's been a wonderful symposium, and one that I know Carl would have loved to hear a lot of the talks. He reached out to me in the early 90s, which is sort of odd. It is true that both of us as undergraduates got our degrees in math and physics. And so I think we thought very similarly, and we're both outsiders in that sense to biology. I went back through all of my emails I've got until, back to the early 90s. And it was interesting to me that he, after we got to know each other, he said, look, we've got to get this ability to do 1 million bases sequencing on the campus. And, could I talk to him about it? And we had he would bring me over to his lab for no good reason other than to tell me about the microbes. And I said, at one point I said, Carl, you are one of the great men of science. Why are you're 20 years older than me. Why do you waste your time talking to somebody who barely understands what a microbe is? And he said, because someday you will figure this out and you'll understand, and you need to know now. And this kind of long-term investment is just mind-boggling. It's rare enough in science today that we do this. Another dear colleague, John Woolley, who we finally lost this year to cancer, and was one of the people who actually started a lot of computational biology in the country, invited Carl to a conference we were having in San Diego back in 2006. Of course, Carl wouldn't come. But in the invitation, which I found dug out in my emails, he felt, and I certainly feel, that it was Carl that really turned me on to biology. And then during the 2000s, I visited Carl here in 2006 and then again in 2009 at their house. So I really have a very deep feeling that Carl actually has fundamentally changed my life. I would talk to Carl and I'd say, Carl, these are just microbes. I mean, I do stuff with big data. Like I wrote 3 papers on different forms of observing the Andromeda galaxy, and there are 100 billion stars in this thing, and there's 100 billion of those galaxies, right? I mean, this is big science, not your bugs. Well, Carl would patiently say, yeah, except, that 100 billion stars times 100 billion galaxies seems like a big number until you think about the number of microbes on this planet, of which there are 100 million times as many as all those stars in the universe. That's a really mind-boggling thing, and I think it's something that we haven't really, because the ability to sequence the microbial ecologies is still in its infancy. We haven't really begun to understand just how vast these numbers are. A few people did. I love this quote from Julian Davies. I got this from Rob Knight. And this was sort of what Carl was too polite to say. But in the end, of course, he's right. Because although there's all those 100 billion stars, they don't have software inside of them that is constantly changing, which means as a dynamic system, the information dynamics alone, much less all the biochemistry, is stunningly more complicated than what I used to calculate in astrophysics. And I spent 25 years doing general relativistic astrophysics. Now, in the late 2000s, when Carl and I would have these half days together, basically, this is from an interview with him in Current Biology, and he was talking about the 2 big directions in biology to come, and that microbial ecology is the most in need of development. both in terms of the framework and in which to interpret them and the facts that are needed to understand them. And oddly enough, this is in 2005, I started working out of never having done biomedical work or had any training in it, in Microbial Ecologies in 2005, because the increase in capabilities of the sequencing had allowed Craig Venter to do his around-the-world voyage sequencing the ocean water. That sequencing was funded by the Moore Foundation, and they called me up and said, we don't have any place to put it. And it's all this data, and we need to make it available. And by the way, everybody else is doing this too. And so there is this beginning, just exponentially growing ability to sequence the full ecologies. And so I said, okay, if you just let me do it my way, we'll do it. And so they gave me a grant, and I built this supercomputer that was dedicated just to microbial ecology genomics. And we began to upload, people from all over the world through the internet could upload what they had sequenced in Yellowstone, hot springs, and in the dirt and in the ocean and so forth. And then it was available to people all over the world. In fact, by the time it ended, there were well over 5,000 users in 90 countries, all say comparing sequencing some local sewage or something to, well, what is this? And then you could go up blast it against essentially everybody in the world putting up their ecologies. And in doing that, I noticed toward the end of that project that these odd eco-niches were beginning to show up. And this one in particular, you'll notice, is human distal gut. And I said, what is that doing in our database? We're supposed to have environmental ecologies. And of course, this was just as the NIH was ramping up the Human Microbiome Project. And so we were beginning to get, you'll notice the second bug is an Archaea. And we were beginning to get a lot of this data. Well, fortunately, the software systems that we had developed for going from the gene sequencing, the Illumina output basically, into the ecology was just as useful for the gut as it was for hot springs. But when you consider the gut, it's really quite an amazing thing. It's the Rodney Dangerfield of organs, right? Who gives the colon any respect? Really? Come on. Heart. If you're a heart surgeon or brain surgeon, man, that's stuff. But it's plumbing, right? Well, in fact, when you begin to look at it, it's the largest immune organ in your body, the large intestine. The density of the microbes is 1,000 times that which we heard Penny describe in the ocean at the start of this conference. It's an incredibly dynamic ecosystem. On hours to days. If you change your diet within a couple of days, you have a totally different ecosystem. And as we know a lot, because they're so jammed together, basically there's between 1 and 100 billion microbes per gram. And so the horizontal gene transfer is of course, a big part of what goes on. There's all kinds of plasmids, viruses, everything else that are helping along that. The phages I won't get into, but I've done a lot of work on the phages as well. But here's the thing. When Carl, remember, he talks about these ecologies, he says you got to think, if you're a biologist and you want to understand evolution, you've got to have the environment and the bugs highly coupled, because evolution is selecting, the environment is giving selection pressure, and then evolution will figure this out. So what about the selection pressures? Well, you've got the active human immune system that is tightly coupled against that microbiome. It has spent its whole life booting itself up as an artificial intelligence learning system on the growing diversity of the microbiome, and it's out there to kill bugs that it doesn't like and not kill them that are there. And it's one of the most amazing things in the world that this thing can tell the difference between hundreds of species of bugs that it assumes are part of you and are okay, and the ones that it doesn't like, or levels of them it doesn't like, that it then activates multiple different components, the innate immune system, like lysozyme, the adaptive immune system, like secretory IgA, and then the white blood cells, the neutrophils, and they shed all kinds of glycoproteins like calprotectin, lactoferrin, and so on. And then the other thing is each little cell is this little incredibly chemical refinery that has these inputs and outputs, and you're constantly changing them with your diet. Antibiotics is like, you're going in and napalming the whole place, and then the pharmaceuticals, God knows what that's doing, right? And so the poor bugs are down there just trying to live, and it's hard. So as I got into this, I won't give you the long version of this, but I decided that the best thing to do was to turn my body into a genomic and biomarker observatory. And so I started taking blood and stool measurements of my body every quarter, then every month. This is on the big 64 million pixel wall at my institute, and each of those is a different, LDL or kidney or the white and red blood cells and so forth, about 150 of them. That's how I gather my data. That's about one blood draw. I don't recommend this, by the way, but as I looked across the data, and I just love those pictures of Carl looking at those spots on those things and almost going blind doing it. I mean, that's what I do. I mean, I have this vast amount of data. It's because you let the data talk to you. You let the data tell you what's going on. And and I found this one variable, which I didn't know what it was. Remember, not only am I not a doctor, I'm not a biologist. But CRP, it turns out, is the complex reactive protein is the general measure of inflammation in your blood. And it's supposed to be less than 1 below that green line. And it was already back when I was talking to Carl. It was 5, 10, 15, eventually 27. To put that in perspective, if it's 5 on average, you quadruple your future chance of heart disease. So it's not good. Okay? But there had to be some source of inflammation inside me, and I couldn't figure out what it was. And the doctors just said I was nuts for doing this. So I, again, felt real solidarity with Carl. I mean, during those early 90s was when some of the dark days, and we spent a lot of time with Carl telling me about how science actually works instead of the storybook version. Anyway, as I kept in my quest, I realized that if I started taking stool samples, I could directly measure the immune system because it's your largest immune organ is shedding into the stool things like lactoferrin. So again, this is supposed to be less than 7, and I'm up at 900. 125 times the upper limit for healthy. You read the literature. So I started reading vast numbers of scientific papers in this area, now 500, 600, something like that. And it turns out that's a sensitive and specific indicator that you have inflammatory bowel disease. Now, I said, really? I'm 60 when I find this out. Like, did I miss the memo? I have an autoimmune disease that's a chronic incurable disease. Like, how did I not know that? And so, and the doctors had never, indicated there was anything. But what you can see is that this is that the neutrophils are attacking something they don't like. What is, when they shed this, it's like tear gas from tanks, to soften up the crowd. This is an antimicrobial glycoprotein which sequesters iron away from the bugs that need it, like Salmonella is an example. It needs a lot of iron. But you notice how episodic it is. Those are years, right? And by the way, these graphs, there are no such graphs in the scientific literature on the human immune system. Why? Because they don't take time series. Here is the adaptive and the innate immune system. And what is the innate immune system? That's lysozyme. It bores holes it's an enzyme that bores holes in the side of gram-positive bacteria, but not so much gram-negative ones, right? So there must be, this must be inducing by environmental time pressure, time changes in the microbiome ecology. Namely, the gram-positive ones that didn't like are getting killed. and they've got to go down. And I ought to be able to see that. I ought to be able to measure that, right? Whereas the secretory IgA is an antibody, and there's a whole realm of these antibodies that are trained to go after, particular things that they don't like. So there had to be a coupled microbiome ecology. Okay, so I decided, okay, well, let's dive in, let's figure out what's happening with the microbiome ecology. Fortunately, the NIH had already done 255 healthy people, and you could download the raw reads from the Illumina sequencers. I had 7 times from like January of 2012 until mid-2013 that I had frozen stool samples so I could sequence those. And then we found in the literature about a couple of ulcerative colitis patients and some ileocrohn's patients. I had colonic Crohn's, which means it's in the large intestine, whereas a lot of the bad Crohn's, ileocrohn's, is in the end of the small intestine, the ileum. Well, this is not 16S, okay, which is about 20,000 Illumina reads or something like this. This is 100 or 200 million Illumina reads per sample. So this is very deep metagenomic sequencing. And the reason was that I wanted to look down into the rarer microbes, but I also wanted to get the protein family distributions as well and see if there were changes in function as well as in taxonomy. Now here's a number I wish I could have talked to Carl about. Remember the 1 million bases he wanted? This one project uses 1 million bases, okay? And so, how do you take that much big data and get anything out of it? Naturally enough, use supercomputers. Now, I happen to be at San Diego, so I use the San Diego Supercomputer Center, is also because it's the only big data machine NSF has funded, and it's set up for this kind of thing. And I also got, Dell gave us a lot of time as well because they wanted to figure out how to get into this game because they see it's going to be a big growth area. But essentially, we used about 25 CPU years. So that's like taking your laptop and running it 24/7 for 25 years. Again, this is something I don't recommend you try at home. Furthermore, Rob Knight had been recruited. I worked assiduously against everybody, probably including you, at Illinois, to get him to come to San Diego and to bring his group from Boulder and set up the wet lab there. But while he was doing that, I said, Rob, as long as you're coming, let's build the best cyber infrastructure under your lab that exists in the world to do with the microbiome. And so we build these, what I call Fionas. They're PCs, but they're optimized for big data. So they would have, for instance, this has 3.5 terabytes of flash memory, and it has 120 gigabytes of RAM and so on. But the main thing is it's connected at 10 gigabits. That's 10,000 megabits a second with a clear channel and no one else on it. So to put that in perspective, in our campus, we have 30,000 users of the shared internet. All of that shared traffic is carried on one 10 gigabit per second optical fiber. So I'm going to give Rob the same amount just for him, right? And we're doing this across the campus for a number of disciplines. That's a Illumina 4000 sequencer, and that we're linking that center up as well. And so that can come into these switches that then can go over to our big wall, or at 120 gigabits per second, 120,000 megabits per second, it gets into the San Diego Supercomputer Center, or we can go out through the networks to NCBI or anywhere else, or for that matter, IGB, which we've discussed adding into this picture. And so the idea is that the big data flow essentially just flows around in this sort of separate big data freeway as your photos you attach to your email interactively flow around in the normal shared internet. So we're essentially in this country at this moment in time building the equivalent of the interstate highway system and the big city freeways for big data. So think of it as like 1955. Now, what happens once you've done all this, and of course you can't read it, but each of those vertical bars is a species The red are Bacteroides. So the first one is Bacteroides vulgaris, and the second one is Bacteroides ovatus, and so on. Then there's the Firmicutes in blue, and then the proteobacteria will come back to in purple, and a few other phyla. So this is organized by phyla, and this is the 75 most abundant species. Normally I do the 200 most abundant species, and I can do 2,500. And that's vertical scale is important, is a log scale. So what you see, what looks like a straight line on semi-log plot, remember, that's a, power law fall off. So what it means is that there are a few bugs, like Bacteroides vulgatus is 10% of all your 100 trillion microbes in a healthy person. This is an average over the 255 healthy individuals. And so we can use the wall to say, look, just look at, in this case, 155 individuals. And from all you need to see is that they're blue and red. And that's what we expect, that most healthy gut microbiome ecologies are basically between, and you saw a graph of this in Rob's talk, are either Bacteroides or Firmicutes. But notice this little one over here. What's up with that? Remember the purple is proteobacteria? And the top, the high mark there is E. coli. So this person is 98% proteobacteria for their ecology. And they're about, I don't know, it looks like about 95% E. coli. Now somehow that person got through the NIH surveys to be considered healthy. And it's in the database for any of you who are downloading what a healthy person looks like. And so this is, again, this sort of thing that Carl taught me is you've got to look at the data in detail, one by one. Don't just let the computer tell you it's right. And look at the difference between these ecologies. Remember, the vertical is a log scale. So you can see that the red, the Bacteroides, is suppressed by about 50 to 1. Now, if you take, the real question is mathematically, okay, so you've got these hundreds of microbes in this ecology. How do I compare one ecology to another one? Are they similar or not? Over all these different species, phyla, and so forth. So using principal component analysis, which is a way to do that, here are all of the 255 in blue, and then the green ones are ulcerative colitis, the red ones, red squares are me, and the pink triangles are ileal Crohn's. And what you can see is that they separate. So what that means is the underlying ecologies are quite different. Well, how different? Well, remember, we've got the average abundance of every single species. By the way, every single strain. I mean, that's the nice thing about metagenomics. It goes way deeper than 16S. You wouldn't be able to tell that. And so if you think about this, look at it essentially as a map. That's the kind of maps that Rob was showing you the movies on. And you'll see that essentially if you're to the left, it's good, you're healthy, and if you're to the right, you're sicker and sicker. And so here I can take my 7 samples and actually look at the time dynamics in those 7 samples over a year and a half. And so my ecology is moving around partly because I've had a month of antibiotics and prednisone during that time. But mostly it's a very unstable dynamic system. Well, at the highest level, if you just look at the phyla, here's what it looks like. The average person, as I said, blue is Bacteroides, and the red is Firmicutes, and the green is Proteobacteria. And you can see that in average, almost 90+% are these 2 phyla. But when you look down at the ileal Crohn's, you see that well, you can't see, okay. That little teeny blue just to the left of the red, that was that 3/4 of Bacteroides. And you can see it's just completely gone. And that big expanse there is Actinobacteria. And if you look over at the ulcerative colitis, which if you were a GI doctor and just done a colonoscope of somebody who you suspected had IBD, it's actually pretty difficult to clinically determine whether the person has, just by looking at the colonoscope, has ulcerative colitis or Crohn's. But you can see that the microbes are completely different. The ecologies, as Carl kept saying, the ecologies is what we've got to look at. So what one of the things I spend, I used to say to Carl, you spend a lot of hours over here just looking at data. And he said, yeah, that's what I do. And I don't know what it is, whether it was Carl or just, I don't know, we were twins separated at birth or something. But anyway, this is what I do. I just love spending hours with the data. So here, for instance, we're looking at families instead of phyla. You can look at anything you want, genus, class, order, whatever. And I've color-coded them by the phyla that they happen to be in. And this is comparing the ileocrones with the healthy. And the question is, what is the disease? What do you got that's making you so horribly sick? Well, the microbial ecology is completely screwed up compared to healthy. And not just that the proteins that it makes, the metabolites, like a third of all the metabolites in your blood, bathing every organ in your body, are made by the bugs. So if you have this incredible change in the kind of bugs that are there, you're going to have all that metabolites, all your organs are going to have to readjust, and that readjust is often called disease. Okay? So, all this is outside of medicine. This is completely outside of medical knowledge, and it's certainly outside of medical treatment today. It's yet to come. It's the medicine of the future. But I thought it was interesting that you can just see here in just this one disease state, the red again are Firmicutes. So the lactobacillus are just like, several hundred times more common, and that probably is correlated with why you have diarrhea all the time. Whereas the Bacteriotes you can see in blue on the left are just incredibly decimated, and so with a lot of the proteobacteria as well. So these are sort of fingerprints, basically, that are, I think, going to be appropriate for a lot of different diseases as we go forward. But it's worse than this. Notice that's like 200 times higher for the most and 300 times lower. So then I just said, let's now take the protein families, and there's lots of ways to do this. This is using the KIG, the Kyoto protein families, and across the bottom you'll see there's 7,000 there's 7,000 dots there, and each one is a protein family. And what I've done is I've taken the average value over these people with ileal Crohn's, and then the average of healthy for each of those 7,000, and then I've divided one by the other. So in other words, how much more of protein 341 is there in the ileal Crohn's compared to in the healthy. And you can see it's on a log scale. And so in this disease state, you have, it looks like 700 different protein families that are greater than 10 times, then there's 100 times, 1,000 times, or 10,000 times overabundant in the gut compared to what is there in the healthy state. And similarly, decreased down to a part in 1,000 of what they are in the healthy state. This is huge. Okay? Now this is the gene content of the microbial ecology. We can of course do transcriptomics to then figure out which genes are being expressed or not, and that's one of the next things we're going to be doing. So one of the things I've been doing is realizing that I can use the wall to look at a lot of these simultaneously, but essentially there are characteristics that you could get with 16S for sure, even things easier than 16S, to figure out what's an E. coli. And you can see here in these ulcerative colitis patients, it's 100 times more than in healthy people. Whereas in so the green, by the way, are ulcerative colitis, the blue are the Crohn's people, and the red are me. Okay, and then the purple, each of these is a person that's healthy. And so here, the CD, which is down here, is 100 times lower than the healthy. And here, the ulcerative colitis, the Crohn's, I'm sorry, is 100 times, let's see, I got that backwards. The Crohn's is 100 times higher than the ulcerative colitis. So by just looking at individual species, you again have essentially a diagnostic as to what the disease is without entering the person's body. No colonoscope, no blood work, just, reuse a little bit of your soiled toilet paper and send it to the gene sequencer. Now, when I saw this, I really had to smile. So Methanobrevibacter smithii is the most common archaea in human gut. It's normally a trace in healthy people, and you can see so many of them, even down at a part in 10 to the 5th, you can't detect it. And somehow I had 18% of all my bugs at my worst point of inflammation were archaea. And as far as I know so far, that's a world record. If anybody knows of anybody who can beat my record, I'd bring it on. And that's why I love Zan's talk, because what has this done? I mean, at the time it was pretty clear that they were producing a lot of methane that I didn't miss, or my friends. But anyway, the point is, there's got to be some hydrogen there, right? And so what are the sub-communities that have been formed in these kind of bizarre states that humans evidently can get into? What are the coupled communities? And that's where a lot of this machine learning that we're beginning to put on this is going to work. So I'll just quickly go through this. This is a time series of those 4 inflammatory markers plus the 3 inflammatory markers plus CRP over time, and those are the pharmaceuticals down there in the solid colors. But you see the bottom, those little green things? Those are the stool samples I've got. So I've only done 7 so far, and I've got 50 altogether. So the next stage is, just last week we were awarded nearly a million CPU hours on Comet, Rob and I, to expand our study, because obviously these are interesting things, but they weren't, you can't do it on just a few people. So we're now going from a few ulcerative colitis to 100 very well phenotyped IBD patients from Bill Sanborn's group, plus another bunch from the Boston study. And then I'm going to all 50 times over the 4 years instead of just 1.5 years, and we're going to do a complete rewrite of the software to Rob's group as well. So I'll just close with the following. What does this mean for the future of medicine? Well, first of all, somebody's going to have to teach doctors and medical students about ecology. They get no training in this, not to mention they don't get any training in microbial ecology. But literally, this is David Relman's work and his team at Stanford. He's writing these papers on what are the things we've learned about dynamics of ecologies over the last 40 or 50 years. And you need to know about this, Mr. Doctor or Mrs. Doctor, if you are going to be actually dealing with humans, who are 90% microbe cells and 10% human cells. Furthermore, that while this focus on individual microbes has been very effective in medicine, and you see on the left that individual bugs that cause disease, the incidence has come way down. But at the same time, that during the last 50 years, all of these modern plagues, autoimmune diseases, diabetes, obesity, asthma, allergies, have increased just as much as the others have decreased. And that's why people like Martin Blaser, who was at one point the infectious disease officer for the United States for the CDC's 40-year veteran, has written a book last year I recommend to everyone on the missing microbes, how the overuse of antibiotics in our society has just caused incredible havoc, along with the fact that a third of our births are C-sections, as you saw what happens then yesterday from Rob. So the whole point about public health is we got to get away from this single microbe focus to, what did Carl tell us? Microbial ecology and its dynamics. That is going to be a gigantic conceptual change. It gets worse because it turns out that it isn't isolated in your gut, but the gut is very heavily coupled to the neuro system and the brain in particular. And so a book that just came out a few months ago, Brain Maker, by David Perlmutter, who I'm working with, MD, goes through the scientific literature and just shows how many neurodegenerative diseases are almost surely started by this kind of dysbiosis in the microbiome, which eventually, as it, pachinko machines through your body, ends up causing Parkinson's, ALS, Alzheimer's, maybe autism. And so this is, again, the fact that the doctors are going to have to become systems thinkers and not organ thinkers is going to take a generation of change. Well, it's a huge cross-disciplinary team that makes this possible. I'll just thank all of those and maybe take a few questions. I would love a fecal transplant, but as a scientist, I'm not going to do it until I can actually sequence deeply the donor, myself before the operation, and then a day after, a week after, 6 months, and so forth. And we're probably within a year of being able to do that. The other thing is you have to be careful. Fecal transplants are incredible. I mean, it's brute force replace. It's what Carl said. Think microbial ecologies. You take a microbial ecology from a healthy person and you put it up into where you have an unhealthy one and let the bugs fight it out, and the healthy ones will win. And that's the reason that if you get C. diff, which is largely induced by the doctors giving you all those antibiotics, and it's maybe 20% treatable by antibiotics, it's 90-95% curative till you have a fecal transplant. We don't know how many other diseases. We don't know, for instance, an autoimmune if you do that because you have a genetic predisposition, which I didn't, I've done my whole human genome. I've got I could spend another hour on the SNPs and so forth that gives you the genetic predisposition to have an autoimmune disease. But the point is you do have that. That hasn't changed. And that, over time, interacting with your microbiome. In other words, those changes in your SNPs are in portions of either the genes or the control region that generate your immune system. And so your immune system is a little off. And that is, of course, the thing that's most tightly coupled to your microbial ecology over your entire life. And so eventually, over time, that presumably has altered your microbiome. And so after the fecal transplant, won't that just start again? So we don't know, but there are clinical trials that are beginning to be underway. And just think how amazing that would be if, but you got to be careful. There was this article recently about a mother who needed a fecal transplant, I guess for C. diff, and she had an obese daughter, and after the transplant she gained 50 pounds because a lot of obesity is tied up in the change in the microbial ecology to a more what's called obese microbiome, because it has a lot to do with the gateway of what you put in your fat cells. And so, you need the phenotype of whoever was the donor as well. It's not as simple as it sounds, but very promising.
How much does a mother's microbiome affect the growing baby embryo?
So it's two things. The mother's microbiome, if you have vaginal birth, for instance, the starter kit for the baby as far as its microbiome is the vaginal microbiome, which within weeks and then days and hours of birth alters itself to the absolute optimal starting kit for the baby, as has been fine-tuned over a million years of evolution. If that is not the way you come out, that's a problem. But there's also epigenetic changes in the mother. So obese mothers not only, there's a reason we have this huge number of obese children. When you're obese at 3, it's not because you've been to McDonald's too many times, right? And so it's both the fact that you're inheriting an already obese microbiome, but also the epigenetic changes in the mother's DNA can be, now it's been shown, passed on as well. And so this is an area, one reason Rob Knight came to UCSD in the pediatrics department is because the first 1,000 days of life, as they say, is where all of this gets played out, and we have so many babies who generously produce samples many times a day. And it's all being collected and frozen and sequenced. And so we're going to understand this a lot better than we know now in the next few years. But that's a good point. It's very central to what we got to get fixed in our society.